Hello, Advanced Biology. I'm really hopeful that this chapter feels really easy. I was really happy when I looked at what we had left and saw that it was stuff that wasn't like wild and crazy and new. So I hope that you see it that way, that we're trying to learn stuff that we've already learned through distance learning / e-learning instead of I already know this, I'm bored. So hopefully you have a positive attitude about it. I know I'm really trying to. Alright, so the study of how characteristics of living organisms are inherited from generation to generation. Since we say study, the answer is not heredity, but rather genetics. So the study of genes and how those cause traits to be passed on. When we're talking about genetics, got its name from this, which is a gene. Okay, so here's my, long DNA strand. It's really a lot longer than this. And I've got those nitrogenous bases in the middle holding the DNA together. So a gene is a certain segment of DNA. So maybe I'm talking like from here to here. All right, it's a certain segment of DNA with information about a trait. Okay, so one example that we can use in humans that's actually, they think, where we can talk about it like it's a simple trait like this, is hitchhiker thumb. Okay, so if you hold up your thumb and it's just straight like that, that is not hitchhiker thumb. But if you extend it, whoop, and your thumb goes beyond straight, that's called hitchhiker thumb. Okay, so we could say this is the gene for thumb shape. And no, I'm not talking about fat or skinny. I'm talking about hitchhiker thumb or not hitchhiker thumb. Okay, but notice I didn't label this gene with hitchhiker thumb or straight thumb, right? This would be this gene for the trait, which is the shape of the thumb. When we're talking about which shape, the form of the gene, then we're talking about an allele. So if this contains instructions for straight thumb, okay, so I would have the straight thumb allele for the gene of thumb shape. Okay, if this gave instructions for the hitchhiker thumb, which I have, okay, then I would have the hitchhiker thumb allele for the gene of thumb shape. Okay, so the gene is the place of DNA for the kind of a category, and the allele is the specific version of the trait that shows up. Okay, so alleles can be represented by letters, depending on whether they are dominant or recessive. I'm going to count that you kind of maybe understand this concept a little bit, but if we're talking about a dominant allele, just like a dominant person, if you've got a really dominant person in the room, you notice them, right? They control the conversation, they make sure everybody's focused on them, right? So that's a dominant person. Same idea with a dominant allele. If there's a dominant allele around, their instructions are the ones that the body listens to. Okay, so they're able to cover up the instructions of a recessive allele if both are present, and we represent them with a capital letter. Okay, so then the opposite of dominant would be recessive, and that would be like a insanely shy person in a conversation, right? So they're not going to talk unless nobody else is talking, right? So if there's a dominant allele, it takes over. But if there is no dominant allele around, then the recessive ones are going to have to step up, and their version of the trait actually gets to show up. So their phenotype is shown only if there are no dominant alleles present, and we use lowercase letters for the recessive alleles. But we're going to use the same letter. Okay, so if I believe hitchhiker's thumb is dominant, okay, so if I go back to my hitchhiker's thumb, remember, so this is hitchhiker's thumb, this would be straight thumb. Hitchhiker's thumb is dominant, so I would use capital H for hitchhiker's thumb, and lowercase h for straight thumb. Right, so it's not like you use an H for hitchhikers and an S for straight. It's the capital and lowercase of the same letter, and we always pick the letter of whichever one is dominant. Okay, I know on the Edpuzzle I signed the other day is an unfortunate where it was blue and brown, so they're both B's. But the capital B was for brown because brown was the dominant one. All right, so this kind of lays out, again, this idea of the gene versus the alleles. Okay, so the gene is this place on every single person's DNA that has instructions about this trait. So it would be the eye color gene. And then the specific instructions in that gene, be they brown eyes, blue eyes, emerald eyes, gray eyes. I know this is in black and white print, so it's not very useful. But those are the alleles. Okay, so this would be actually more complicated. I don't only have 2 alleles, I have 4 alleles. Okay, but we'll talk about how those work in a little bit. But the idea is we have this place of DNA with instructions is the gene, and then specifically what are the instructions for? Those are the alleles. Okay, and we would pick some letters to represent each of those alleles. Same idea with hair color. Okay, so this piece of DNA with instructions about hair color is the gene. The specific instructions that are on the gene could be for blonde hair, for red hair, for brown hair, for black hair. And both of these are really more complicated than that, but it kind of hopefully is driving home the idea of a gene is a place in the DNA, and the alleles are the really, really specific instructions of that gene. All right, so quick rehash here. Sex cells, those are the gametes. And remember, gametes have only one copy of a gene, and when we represent this with just an N, N's like for the number of sets of information. They have one set of information, which is half the amount normal cells have. So those are haploid. And then body cells, this is maybe a new word. Any cell in your body besides your gametes are called somatic cells. Or body cells, somatic cells. And they have 2 copies of everything, one from your mom and one from your dad. So they represented 2N. They have 2 copies of all the chromosomes. We call them diploid or diploid. And so how do we get from haploid gametes to diploid, basically your entire body, except the gametes that you make when you hit puberty? Well, the gametes come together, they fertilize. And so we have a haploid egg and a haploid sperm, and we come together. N plus N would be 2N. So they generate an organism's first cell, which again, remember, is the zygote. When I pause like that, I'm hoping that in your brain you're trying to remember the answer, but I don't know. All right, page 3. Maybe. Yes. Okay, this is possibly a new word for you. When we're talking about a species, okay, we can talk about all of the DNA that a human has versus all of the DNA in a mosquito versus all of the DNA in a cat. And we call all of the DNA for an entire organism its genome. Okay, mapping the human genome was a huge project, and it was a big accomplishment. I think they finished it in the late 90s, but they figured out basically of all the 46 chromosomes in a human, 23 pair, how many genes are there, and what does each gene do. And they definitely figured out the sequence of the genes, but they don't necessarily know what each gene does yet. So they have the sequence of the DNA, but they don't quite have it all figured out what does what. But we're going to generally focus on human beings in here. So our human genome is 23 pair of chromosomes. And I want you to think about when you've seen all the pictures of chromosomes, right? So here's a chromosome with its copy, and here's a chromosome. All these chromosomes. How many base pairs of DNA is that? So remember, if we drew DNA I'll draw it again here. Right, so we've got those base pairs, so I have like C and G and A and T, and I know that's a little small, but remember, there's all these base pairs. The pairs make up the inside of the DNA. Okay, so if you think about it, in all of the chromosomes of one cell of one human being, again, this is in one cell, of which you're made of like 1 billion cells. Okay, there are over 3 billion base pairs in one cell of one human being. Okay, so that's an insanely high number of base pairs. Okay, of those base pairs, okay, so how many proteins do you think that can code for? Remember, a gene is transcribed into mRNA, and mRNA goes out to the ribosome, and we make proteins. Remember, DNA is instructions for proteins. So in 3 billion base pairs, in 23 pair of chromosomes, how many genes do you think we have? Well, we have like 20,000. Okay, so your body's capable of making at least 20,000 different genes. Or it has 20,000 different genes, which means it can make 20,000 different proteins. So it's really just an overwhelmingly large amount of stuff. So again, when we talk about, we always say we have 23 pair of chromosomes. Remember where those come from. Okay, they come from one from each of your parents. So my mom and dad are Linda and Kermit, and I have 23 chromosomes from mom, 23 chromosomes from dad, and they're each numbered, like geneticists like to number them 1 through 22 plus the sex chromosomes. So 1 through 22, my mom and dad's chromosomes that they gave me are basically identical. Okay, so my dad gave me a chromosome number 1, and my mom gave me a chromosome number 1. And they have the same genes in the same places. They're basically the same size. And so those are the ones we call homologous chromosomes. Okay, they're the same size. They have the same genes in the same places. Remember, homo means same. So the alleles for an organism, the alleles an organism has for a certain trait, okay, are what we call its genotype. Oops. While the physical trait that it presents in an organism is phenotype. Okay, so if we go back to the hitchhiker thumb idea, so I'm pretty sure my mom has a straight thumb and my dad has a hitchhiker's thumb. So I have a hitchhiker's thumb. Okay, so I must have the genotype with at least one dominant H allele for hitchhiker's thumb. But my mom has a straight thumb. Straight thumb's recessive, which means she didn't have any of these. So could I have got one from her? No. Right? The only allele I could have got from my mom, if she has a straight thumb, is this one. Okay, so my genotype for hitchhiker's thumb must be this, because I have hitchhiker's thumb. I know I have at least one dominant allele, but since my mom must not have had a dominant allele, I must have got that one. So this is on one chromosome that I got from dad, and this is on another chromosome that I got from mom. This would be the genotype, okay, the letters of the alleles that they have. And the phenotype would be what shows up. I don't know if I spelled that right. So the phenotype is physically what trait does that organism have. All right, so genotypes where both alleles are the same, identical. Again, homo means same. We call them homozygous. Or if you were referring to them, you could say they are a homozygote. Okay, but I usually use homozygous. And then if they're different, instead of homozygous, they would be heterozygous. All right, so we have Gregor Mendel, which we spent a lot of time talking about in normal biology. We're just going to kind of footnote him this year. So Gregor Mendel, an Austrian monk, he did lots of experiments on pea plants, but in doing so, he came up with what are called the 3 laws of inheritance. And I think we're going to get through these 3 laws of inheritance and we're going to stop. So the first one is the law of dominance. Okay, so this is basically just saying what the idea of a dominant allele is. It says dominant versions of traits only need one allele to show up, while recessive versions of alleles only show their version if there are only recessive alleles around. So remember, think of recessive alleles like really, really shy people. They only talk if they're only with other really shy people. Okay, so if there is a dominant allele around, they control the story. All right, so that's the law of dominance. Next one, okay, is going to be the law of segregation. Okay, so segregation means to separate. Okay, to keep separate from each other. So this says during meiosis, alleles are separated and put into different gametes. So we talked about meiosis till we were blue in the face. All right, so remember in meiosis, let me draw a really simple chromosome here. So if I had, I'm going to draw a 2 chromosome cell. Okay, so there's my chromosome from mom. There's my chromosome from dad. So this would be a kind of some organism that has only 1 pair of chromosomes, right? So at some point, okay, in interphase, those chromosomes would copy. So then they would have their copy. Okay, so remember this is interphase. When we copy our DNA. Oh, it froze up. There we go. Then I'm going to leave crossing over out for the purposes of this. I'm just kind of trying to keep it simple. In meiosis 1, our 2 chromosomes are separated from each other. So one cell would have the one from one parent, and the other cell would have the one from the other parent. And then we have meiosis 2, and my cells are going to have to start getting smaller. Okay, where we take this chromosome and we split it up. We take it apart from its copy. Sorry, I still do sound effects even though I'm just alone in my kitchen. So these chromosomes that were together have been separated. Okay, and if you look of my gametes, 50% of them have the one I got from one parent, 2 of them out of the 4, and 50% have what I got from the other parent. Okay, so that's this whole idea. So let's say maybe this had the gene for hitchhiker's thumb, or the allele for hitchhiker's thumb, and this had the allele for straight thumb. Okay, so then in my gametes that I create, 50% of them get the allele for hitchhiker's thumb, and 50% don't. Okay, that's the law of segregation, that these alleles that are, together in the cell, I would write my genotype like this. I would write my genotype with them together. In meiosis, in the process of making gametes, they get separated and put into different gametes. All right, I'm going to do this last law, and then we're going to stop. So the last law is the law of independent assortment. Okay, so law of independent assortment says, okay, if I have more than one gene, okay, well, I'm just going to draw metaphase 1, maybe, of meiosis here. Okay, so remember I'm using red and black to be the chromosomes I got from my 2 different parents. Have to put more than one chromosome on here, it doesn't make any sense. Okay, so this is metaphase 1. Okay, so remember, they're going to be pulled apart. This one's going to go this way. This one's going to go this way. So in this particular version of my metaphase 1, the red one will say red's from dad, gets pulled this way, and the black one I got from my mom is going this way. Okay, down here, it was the other way. Okay, the one from mom went this way, and the one from dad went this way. But if I did this meiosis again to make more eggs, it could have been the opposite. Okay, maybe both the red ones go to the left, or both the black ones go to the right, or the opposite. Okay, so which way, so for this pair, whichever way they go, red this way or black this way or whatever, does not affect at all what happens down here. Okay, this chromosome could go this way, this chromosome could go this way. It does not matter what this one did. Okay, these chromosomes are doing their own thing. These chromosomes are doing their own thing. They assort independently of each other. Okay, so the passing of one allele, maybe there's an allele here that I'm studying. Okay, so what happens to this allele does not affect what happens to some allele on some different chromosome. Okay, they're independent of each other. They each have their own separate probability. We're going to save the old Punnett square conversation for a later video. See you guys.